The central dogma of molecular biology describes the process of taking DNA and converting it into other types of genetic information. There are three major steps that help compose central dogma: replication, transcription, and translation. In replication, the DNA is unzipped and replicated into an old and new copy of DNA. In eukaryotes, the original copy of DNA remains in the nucleus, while the new copy is free to leave through nuclear pores to be used elsewhere within the cell. In prokaryotes, replication occurs in the cytoplasm. In transcription, this newly copied DNA is transcribed into RNA with the help of RNA polymerase. There are different types of RNA, but mRNA specifically is what is used to produce proteins in what is called translation. However, before we get to that, we'll be mentioning an exception to the transcription process. Transcription can also go in reverse in what is called reverse transcription. Reverse transcription is when RNA is converted into DNA. Retroviruses are notorious for using reverse transcription in order to insert their genetic material into the host DNA. This allows them to make countless copies of themselves in order to infect the host. Lastly, we will look at translation. In translation, the mRNA strand is translated into a protein with the help of tRNA, which brings amino acids to help create the primary structure of a protein. This primary structure is a chain, but when it interacts with other chains, will create the quaternary or 3D structure of a protein. To help think of it better, translation is the process of taking a nucleotide sequence, RNA, and converting it into an amino acid chain sequence, eventually a protein. That being said, RNA is not always converted into proteins. As stated before, there are other types of RNA. mRNA is the only one that is used to create the protein structure. tRNA, rRNA, and snRNA are all used to help produce it, but are not used as templates for them. As of this moment, reverse translation does not happen biologically due to certain genes not being expressed in every single protein.